In this section, we will talk about reverse transcriptase and allele-specific polymerase chain reaction. But before starting this section, let's discuss the question asked from you in our previous section. That was, what do you know about primers for polymerase chain reaction? Two sets of primers are used, which are short stretches of oligonucleotides. They are chemically synthesized and complementary to the part of the deoxyribonucleic acid template to be amplified. Deoxyribonucleic acid polymerase requires a primer for initiating replication. Deoxyribonucleic acid polymerase cannot initiate the process. It can only add nucleotides. Therefore, a primer is used for this purpose to initiate the polymerization process. Primers are usually 20 nucleotides long. The two primers used cover the target region of deoxyribonucleic acid to be amplified and bind to the opposite edges of the template strand. After binding of primers, the region between them gets copied by the deoxyribonucleic acid polymerase. Now moving towards our topic, and that is reverse transcriptase polymerase chain reaction. The reverse polymerase chain reaction approach uses messenger ribonucleic acid as starting material. After the first primer is attached, complementary new deoxyribonucleic acid is synthesized by reverse transcription. This is used as a template for a new deoxyribonucleic acid strand. Then the second primer is attached. Subsequently, multiple copies of complementary deoxyribonucleic acid are produced by polymerase chain reaction. Now let's discuss the allele-specific polymerase chain reaction. This amplifies a DNA sequence from one allele only, and not the other if it is different. If allele 1 contains adenine and thymine base pair at a particular site, and allele 2 contains cytosine and guanine pair, now allele-specific polymerase chain reaction is normal. For allele 1, if a mutation has changed in allele 1, the thymine by a cytosine, amplification is not possible. Similarly, polymerase chain reaction for allele 2 will be normal, but not if a cytosine has been replaced by adenine. In allele 2, allele-specific amplification is not possible. So, before ending our video on deoxyribonucleic acid amplification through polymerase chain reaction, let's revise main steps involved in polymerase chain reaction. So, the first step is denaturing deoxyribonucleic acid into separate stands. The sample is heated to approximately 95 degrees Celsius for 1 minute to separate the deoxyribonucleic acid into single strands. Now moving towards second step of polymerase chain reaction, which is annealing primers to flanking regions of single strands deoxyribonucleic acid. The sample is cooled to approximately 50 degrees Celsius for 45 seconds. As we have discussed, after denaturation, single strands deoxyribonucleic acid will be formed. Two primers anneal to a complementary sequence on the single strands deoxyribonucleic acid. Moving towards third step, and that's extending primers with deoxyribonucleic acid polymerase. The sample temperature is raised to approximately 72 degrees Celsius for 2 minutes to initiate the synthesis of two new strands complementary to the original deoxyribonucleic acid strands. Deoxyribonucleic acid polymerase adds nucleotides to the 3' hydroxyl end of the primer. So this was all. Stay tuned to sqadia.com for more precise and knowledge-stuffed videos.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.